June. Okay, so I hit the record button. Chapter 3 it is. We are going to be looking at these PowerPoints. You have them as well in your, like I said, I just put them up. I'm going to do this as well, I think. All right. So I just made this generic. Welcome to Chapter 3. This is for the 6th edition of the book, which most of you have. Bear in mind, I still do not. I'm hoping to get it sometime this week. Only version I have, like many of you, is the online version. You guys are aware that you can, and I'm sure you've been contacted, you may have been contacted by McGraw-Hill for like 25 bucks, you can get an actual physical copy of the book. It is a 3-hole punch version, but they'll send it right to your house. So you're also welcome to do that. So again, let's get started with Chapter 3. The beginning of Chapter 3, the whole chapter is called sort of like the tools of the trade, or the tools of the microbiology laboratory, because we're trying to get you introduced to how microbiology gets accomplished in a hospital or in a research setting. And one of the opening pictures is this one. It's called A Guide to the 5 Eyes. They are called the 5 Eyes because they all begin with the letter I. That's supposed to make it easy for us to remember. Myself makes it difficult, but nonetheless. Prior to the 5 Eyes, notice that I've put a specimen must be collected, and the sources could include all kinds of things. I included clinical things in this list here, an open wound, a urine sample, someone may draw blood, or a biopsy fluid from a tumor, or I forgot to put cerebrospinal fluid. That would be also a fluid that could be drawn as a sample. And if we're thinking about in the research setting, of course, these specimens might be collected from someplace like the environment, or like soil or water, or maybe if you're testing sewage waste water, that would be the source of the specimen collected. If you're testing pasteurization or something in food microbiology or drug manufacture, obviously there are steps along the way where specimens would be collected to see if the preparation has been contaminated with bacterial or fungus during the manufacturing process. Please note, right, it's required that you can list and define each of these 5 Eyes, but you are not required all of the details. The details of section 3.1 we're going to include when we get to chapter 7, because most of these details include things we're going to learn in lab, as well as things that have to do with how you feed microbiology, how you feed microbes. So again, you need to be able to recite the 5 Eyes, you need to know definitions for them, or a general terminology for what they mean and what is accomplished during each of the 5 Eye steps. But the nitty-gritty details of different types of media and different types of what selective media is versus, you do not need to know that at this time, okay? So let's just run through these kind of quickly. The first of the 5 Eyes we call inoculation, and this involves taking a sample from the source, of course, and putting it into sterile growth medium. Now, this sterile growth medium, I would like to mention, let's see if I can just minimize this. No, you can't do that. All right. So this growth medium could be a liquid, it could be a broth. That's what we call liquid media. It could be a solid media. You see a person here putting bacteria on a solid media. Notice she's wearing gloves, and this instrument that she's using to transfer the media would have to be sterile. And what that means is that there's no living organisms on it, as well as the media itself. It needs to be sterile before it's being used. So the first thing happens, you take a source, you get a source of bacteria, and you inoculate them onto some media where they will be happy and grow. Then you put them in an incubator. This creates proper growth conditions. You can see the person here fully clothed as if they're in like a P3 facility, and they are putting some samples into the incubator. Incubators, they look anywhere from, they can be desktop size, looking like a small college refrigerator, to like a big, usually they have a window in them so you can see what's inside the incubator, and they're held at a particular temperature. Usually If we're studying human bacteria, for example, we're looking at human pathogens, then we would hold that incubator close to 37 degrees Celsius because that's your body temperature. So usually they're between 35 and 37. They have a thermostat on them and they turn on and off and stay very close to that temperature. Bacteria grow super quickly. Most bacteria multiply within between 20 minutes and an hour. They duplicate their numbers. So the time in the incubator can be very short. Sometimes it's even one person's shift, or certainly if you're in a clinical laboratory, by the time you come back the next day, it would be time to take those specimens out and take a look at them. Now, if we're studying bacteria that live in volcanic vents, of course, and the temperature there is 95 degrees Celsius, we would have to construct something or buy something that could hold that temperature. Likewise, if we're studying bacteria that grow in the refrigerator, we would want to incubate them at those temperatures. So that incubation temperature of 37 degrees is particular for humans and other animals that are close to that body temperature. Now, when you sample a wound or a site, usually there are more than one bacteria in that site. And so the third eye, so we have inoculation, incubation, that third eye we call isolation because we are going to separate the bacteria from each other. And this example here is what we call a streak plate or streaking for isolation. And what I need you to notice here is these areas where there's all this, where you see all this whitish, yellowish fuzz, that's thousands and thousands and thousands of bacteria colonies. These out here, where they're just single all by themselves, those would be an individual culture, and we would consider that an individual colony. That little dot originated from one bacteria that multiplied in the incubator. Normally, then, these are put into subculture so that they don't get contaminated. If we would have more than one thing on this plate, different colors, different shapes, different sizes, we put them in subculture, incubate them again so that we would have individual pure cultures for each species that were in our original sample. That's what we mean by isolate, to separate. Then this next one is inspection, and it's very broad. Inspection includes both macroscopically, meaning what you can just see when you look at the colony. And I know many of you might think, well, they're so small, what can you see? Well, you can tell the color, you can tell the shape. You're going to see that in the lab, we actually look for the edges, what the margin looks like, if they're raised or if they're flat. There's all kinds of characteristics that you can see. And sometimes, once you've done this many, many times, macroscopically does give you a good clue as to what kind of bacteria it might be. And then microscopically, which would include, of course, staining. It says possibility. Yeah, you can't see most bacteria without staining. You can see a few fungus, but not any bacteria. So it would involve staining and then looking at them under the microscope. And we consider that still the inspection portion, not the identification portion, because under the microscope, we can't tell any of their biochemistry. All we can tell is their shape, which we call morphology, and we can tell their arrangement. And sometimes we can tell a little bit about their cell wall, but that's it. We can't tell how they make ATP, or if they will digest protein, or if they'll only eat glucose or only eat lactose. That's why it's still in the inspection stage, the microscope portion, because from the microscope, usually you cannot completely identify an organism. Identification comes at the end, and notice it specifies down to the species level, and they're showing you a DNA gel here, results of some gel electrophoresis. So this implies that they've actually looked at either the DNA nucleotides that a particular organism has. And for our purposes and for the purposes of most microbiology clinical laboratories, many of the tests can just be biochemical tests, and we can figure out the answer. We can figure out what the name of the organism is based on its biochemistry. That means mostly its metabolism. Again, does it eat specific sugars? Does it eat specific fats? Does it have a particular enzyme? And we can test for many of those things. Immunologic testing, that means looking for antibodies, as well as sometimes looking for antigens that a particular bacteria may have on its surface. And then, of course, genetic testing, if we need to know specifically an organism that's hard to find, or we can only identify it this way. In a hospital laboratory, in a clinical laboratory, primarily these biochemical tests for microbes, particularly for bacteria, are going to be the first round of tests. And then if that doesn't come up with any answers as far as identification, or if it is somewhat nebulous, then some of these other methods might be added on. So this picture, this is one of the next panels in your book, and this is, of course, to remind us the scale of microbiology. Microbiology, right? Biology, study of life. Microbiology, study of small life. So you'll see here that this is about the range of a human eye. You can see things down to about 1 millimeter. Smaller than that, like 100 micrometers, you're going to need a microscope. And this is showing you here the range of a compound bright field microscope like we will use in the laboratory. And that's going to be between 100 micrometers and down to 1 micrometer. That's a little ambitious. These E. coli will look very, very tiny under a compound bright field microscope. Most of you will have difficulty telling me whether they are rod-shaped or bacillus-shaped or they're round-shaped. Notice that the Giardia here, this is a whole single-celled organism, a protozoan, a eukaryote. Here's a picture of a red blood cell, yeast, and then bacteria. Notice the bacteria fall mostly in this category between 10 and 1 micrometers. Smaller than that, viruses, we would need to use an electron microscope. And you can see this gentleman sitting here in front of an electron microscope. And as you can tell, it's a much bigger process to prepare the specimens for the electron microscope, as well as you don't just like throw them under the microscope and look at them, right? These microscopes are usually limited to research situations. They're not usually used for clinical identifications. And then there are some special microscopes that we call scanning tunneling microscopes and atomic force microscopes, and they can actually see down to individual atoms and very small molecules. I would remind you, this is the symbol for micrometer, right? It's the Greek symbol, lowercase for mu, and the M for meter. A micrometer is 1/1000th, 10 to the minus 3 of a millimeter, okay? A nanometer, 10 to the minus, so a micrometer is 1 times 10 to the minus 6th of a meter. A nanometer is 10 to the minus 9, and I think an angstrom is 10 to the minus 12, right? It's 0.1 micrometers. It's very, very tiny. Picture of the compound bright field microscope. Since this is the most commonly used microscope in the laboratory, this is if you're in vet tech, for example, or maybe if you're working in a field, sort of a field situation, you may actually use a microscope that looks kind of like this. I would expect you to know the basic parts of the microscope. You should be able to identify the ocular, the objective lenses here that change. There are 4 of them on this microscope, where we call this the rheostat. This is the light intensity control. It's called the rheostat. Mechanical stage, to be able to control the aperture, and then the focusing knobs, the coarse focus and the fine focus. I would remind you from Bio 1 that we figure out the total magnification of a specimen based on the power of the ocular lens multiplied by the objective lens that's in use. So in this particular case, which this lens has the red band on it, so I know that's one of the smaller lenses. So for example, oculars are usually 10x. Notice that the 10x is filled in for all of these. If I'm on the 10x objective, the total magnification is the power of that objective lens times the power of the ocular lens to give me a total magnification of 100x. What you will see is always in books and what we will expect in lab is that you report the total magnification only. You don't report 10x times 10x, you just write 100x, okay? The very best super duper compound bright field microscope can have a 200x oil immersion lens, and then it would magnify 2,000 times the size of life. The microscopes, even if we were using the microscopes in the laboratory, they only magnify 1,000 times the size of life, and I have no idea the microscopes that you're going to get in your kit. I think they're in this 400 to 600 range, actually. Here's a person using a microscope. All right, so I'm showing you this because I want you to follow the path of the light rays. Right, so how they're getting up to her eye. Notice we've got a light source. It's being, the condenser's job is to focus that light source, and then so that most of the light gets through the specimen, and then through the magnifying of the objective lens, a mirror, and then another lens of the ocular lens, right? Now, what happens is as the lenses get larger and larger, they have more curvature in them. And because of that curvature, there is some airspace that does not keep the light focused from the condenser all the way through, maximized to the person's eyeball who's looking at this. So, and the reason for that is because when there is an air pocket between the lens, the glass of the lens, and the glass of the slide, air is very refractive. So when the light travels through here, it bounces out and you don't get very much light that's actually continuing on to your eye. And so things look blurry and they're dark and it's hard to see. And so in the 1800s, when microscopes, compound microscopes were much more available, were just becoming available, I'd say like the mid 1800s, 1850s, 1860s, they very early on figured out we have to have something we could prevent this loss of light and this bending of the light rays when it hits this air pocket. And so immersion oil was invented, and that's what it's called, immersion oil. The procedure to use it is usually referred to as oil immersion technique or using the oil immersion lens. And it is only for the 100 or 200x objective. And what you do is you place a tiny drop of this very viscous oil in between on your slide before you slide that 100 or 200x objective into place, and it fills the gap. See how in this right-hand panel here, you see how this is all filled with oil and this still has the air pocket. This oil is specially manufactured so it has the same refractive index as the glass in the lens of this microscope, in this objective. And literally on the lens itself, it'll tell you what refractive index of oil you need to purchase. And then on the bottle of oil, if it matches, then it's going to be a perfect match. And you put a drop of oil on there, and then that prevents this air gap, this refraction, and you get a better picture of what you're actually taking a look at. Now, even using that, there is a limit, which we call the resolving power of a lens. There is a limit to how clearly you can see something as it gets magnified. And this is just an example that was in your book. And I put it in here because for two reasons. One, because it actually shows the limit of the resolving power of a compound bright field microscope, which is 0.2 micrometers. And if something is smaller than 0.2 micrometers, what happens is you can't resolve it using a compound bright field microscope. See how these two bacterial cells are separate in this picture? Because that's how they really are. But under the microscope, because the resolution is only down to 0.2 micrometers, if they aren't 0.2 micrometers away, we just see them as one thing. Same with this eukaryotic cell. You can see some of the internal features of the cell are put here in reality, but we can't see them with just a compound bright field microscope. So to give you a little perspective of this, I chose this picture of the paramecium. I like that your textbook does this. It keeps the paramecium and using several different microscopes, and it shows what the paramecium would look like, stained and not stained. I'm just going to run through these rather quickly. My purpose for what you need to know about these microscopes is that you should know the maximum magnification. And that's pretty simple because all of these you'll see on this particular panel, they're all versions of a compound microscope. So their magnification, the maximum is always the same, 2000. The resolution is the same. It's just changing the perception and changing the filters basically of what we can, so we can see more detail. This is a picture of a paramecium, which is a protist covered with cilia. You may remember them from Bio 1. And this is stained because on a bright field microscope, because the field of view is bright, when you look in there, it's bright white background. If you put something clear on a bright white background, you can't see anything. That's why we have to stain them. The disadvantage though is that all kinds of staining always do some damage to the cell. They always alter a little bit how we see them or what we can see. So here's our paramecium friend stained with a compound bright field microscope. And then here he is under a dark field microscope. And I apologize, I didn't include that panel here because it's not one of my particular favorites. And dark field microscopy is not used as much anymore because we have something better, right? The key factors for dark field is that when you look in, the field is black or dark, you don't see anything. There's a special attachment to the condenser, and you only actually are seeing light that's reflected off of the specimen itself. So the specimen looks bright, and the advantage of a dark field scope is that you can look at live specimens and you don't have to stain them. So if it's something very fragile that might be distorted or ruined, if you stain it, you might be able to use a dark field microscope. But better than that is this picture here. This is a picture of what we look at our same paramecium. This microscope is called a phase contrast microscope. And it is called that because it can polarize the light, much like sunglasses can do, or the windshield of your car, or if any of you used an old school camera and had a polarizing attachment, a polarizer that you attach to the lens of your camera so you could take pictures in bright light. That's what it does. It crosses the wavelengths of light so that you can see more clearly. So these phase contrast microscopes, they're almost like a dark field and a bright field combined, in that this specimen isn't stained, and you can actually see even more of the details of the edges and inside the cell. See how much more clearly the inside of the cell is compared to this guy? Right, we might be able to make out a few features inside, but look at all of the stuff we can see with the phase contrast microscope. Notice the magnification is the same. It's only 230, so it's a 23x objective with a 10x ocular lens. So it's 230 times the size of the light. As I mentioned, this phase contrast microscope, it's a little more expensive, but most, I would say almost all research labs would have them. All hospital labs have them as well, clinical labs, and it sort of makes the dark field microscope obsolete. The phase contrast microscopes cost maybe 2.5 times a compound bright field microscope, but you can just change the condenser, and sometimes you can actually change the light source and also use them for fluorescent microscopy, which is down at this bottom panel. And so it makes it a lot more cost effective. We're going to skip differential interference because it's not used very often. And we're going to jump down here to fluorescent microscopy. And I'm going to see if I can make that picture a little bigger, maybe. Fluorescent microscopy is the same as, it's basically a compound microscope. I said it's usually a phase contrast microscope. And basically what happens is the light source is changed out from white light or a light bulb to fluorescent light, so ultraviolet light. And the stains that you use are fluorescent dyes. And so they will glow under UV light. So these are some cells here. And you can stain different parts of the cell. You notice there's 3 colors on here. There's a red, a green, and a blue. You can either stain different parts of a single cell, or you could stain mammalian cells one color and a particular bacteria a different color, so you can see the interface between them. So it can give you some more information. One of the other challenges though with fluorescent microscopy is you do need to view the specimen in a completely dark room, like almost like a black dark room, right? Like as if you were taking photographs or developing film, which can be difficult to have a space like that inside a main laboratory. If we want to see larger things, one more type of compound microscope, it's called a confocal microscope. And I regret not including this panel in the larger blobs over here. Notice it still has the same magnification and the same resolution. The advantage with the confocal is instead of using ultraviolet light, UV light, it actually uses laser light. You get a bigger range of fluorescent dyes that you can use to stain things. And for confocal microscopy, oftentimes it's used to watch time-lapsed or take video of a process inside a cell. So for example, those of you that have already had anatomy and physiology and you know about muscle contractions, and you know about how the myosin head grabs on to the actin filaments and it pulls and the ATP is released. How we know that in sequence is because those different items have been stained and confocal microscopy, and you can actually watch the process. Now, if you want to see things that are smaller and with better resolution, you need to use a different kind of microscope. So you need to switch from using the wave portion of light to the photon or electron beam portion of light. So you're going to use an electron microscope. And we have two major types of electron microscopes, the transmission and the scanning. And I highlighted the differences between these two by showing you two different things. I thought we have to put the coronavirus in because of all of the distraught that it's causing us, right? This is not COVID-19. This is probably SARS virus. Notice the magnification is 100,000 times the size of life, not 200 times the size of life, 100,000 times. And what I want you to notice over here on the table is that that magnification can be as much as 1 billion times the size of life. Notice the resolution is 0.5 nanometer. So now we not only can see things that are much smaller, but we can also see them with much greater resolution and tell much greater things about the features of a particular structure. The challenge with electron microscopes is that always the organism is embedded in a solid, and then it is sliced. And so you are always looking at a cross section or a slice. You're taking a look through a particular cell or organism in this case. For viruses, this is the only way we can physically see them, is using an electron microscope. The scanning electron microscope, this is some algae, this is a diatom, and all of these little calcium discs make up the surface of the diatom, and you notice this is also teeny tiny. It's only magnified 10,000 times the size of life, so you can tell that diatoms are much larger than viruses. But to see this kind of detail, we could never see that using a light microscope. Okay. Scanning to see the outside, usually it's a very three-dimensional picture. Transmission, a slice or a section, and we can see inside the cells or the organism. I'm just going to mention these other two types here, the atomic force and the scanning tunneling. You might remember that in this diagram here, these are the types of microscopes that we can use to look at DNA and amino acids and even atoms themselves, which is kind of crazy. All right, so now on to, or back to, the compound bright field microscope, this microscope. Remember, in order to see things with this microscope, because cells are clear, we have to stain them some way. So the last little section of this chapter is about preparing specimens for staining, okay? So this here is very procedure and protocol-ish. Again, if you receive your kits, I believe you're going to go through something very similar with the gram stain, but we do need to talk about staining and how it's used in order to help us see bacteria under the microscope. So of course, thinking back to the 5 eyes from the beginning of the lecture, right? We would be collecting the specimen, inoculating, perhaps incubating and isolating, and this is the inspection step where we're going to actually do the staining, okay? So in our laboratories, we would just give you a culture, a pure culture of bacteria. So you would already be to this step. You wouldn't have to collect the specimen or isolate the specimen from different bacteria. You'd already be right here where it says make a smear. And it's very simple. You just put a drop of water on a slide, and then with a sterile loop or a needle, you would sterilize them in a burning flame from a Bunsen burner. You would transfer a tiny portion of the bacteria into the water, mix it around, and then we call that a smear. Okay, that's why it's called make a smear. It's not super technical. It's take some bacteria and smear them around in some water until it's slightly cloudy, and then we say, okay, that's a smear. So you make the smear, then it needs to be dried, and the best way to dry it would just be to let it sit on the bench until it dried. That would be the most least amount of distortion that you would get. However, in the lab, we're usually in a little bit more of a hurry, so we would put them on what we call a slide warmer, and it's kind of like a hot plate with a cover over it, so that we can heat things up with pretty quickly, but not so fast that we cause them to burn or explode, the cells to explode. After the smear is dried, you need to fix it. And what this means is, and I should have put this on here, this means is it does two things. I put the two methods you can use, either heat or alcohol. But underneath that, I should put used to cause the bacteria to adhere or stick to the glass of the slide. And the other important feature of fixing is that it also kills any bacteria that were not killed by the fixing process. Oh, sorry, by the drying, not killed by drying. And that's important because if we're in a clinical laboratory and we're dealing with things that are potentially, that can give us an infection, we would want them to be all dead before we looked at them, right? Then you apply the stain, and you usually do that, it's usually in a dropper bottle. You put a few drops on, and then that's it. It just, stains. You put it on for like a minute, and then usually you wash it off with water. All right, so that's better. The purpose of this panel here, which in the 5th edition of the book was table 3.8. Actually, I think that I got this from the 6th edition from the PowerPoint. So this should be current with your textbook. The purpose of showing you this is to compare positive and negative staining, and that has nothing to do with the charge of the bacteria. Okay, it has to do with whether or not the bacteria have a color or whether the background is colorless, has a color, and the bacteria is colorless. Both of these that I'm showing you over here on the side, both of these are a single dye, one or one stain, and so we refer to them as simple stains. But a simple stain could be a positive simple stain or a negative simple stain. The positive simple stain, the bacteria have a color. See how you can see here these bacteria with flagella, they're little purplish blue rods. Here are some dyes that are frequently used for a simple stain. This is a negative stain. Notice that these are the bacteria, the clear rods and these little white circles, the background is stained. And we use this brownish stain called nigrosin. Excuse me, but this actually looks to me more like it's India ink, which is just like ink that people would use for calligraphy, basically. It's kind of bluish purple, and the background turns that color, and then the bacteria themselves are clear. Now, it gets a little more complicated. There are other types of stains, okay? So remember I told you one stain, one dye, and they can either be a positive stain or a negative stain. So it can either show, let's go back to this one. It can either show the bacteria themselves having a color, that would be a positive simple stain, or the background being a color. Now, why is that? It has to do with the charge on the surface of the bacteria. All bacteria, almost all bacteria, have the cell wall, which is their outside covering, and it is slightly negatively charged. So any stain that is overall positively charged will stick to it. Bacteria is negatively charged, stain positively charged, opposites attract, you get a color. If I use a dye that is negatively charged, then it'll be repelled. So a negative stain, it's negative color and negative on the cell wall, right? So negative to negative repels, so we get the background colored, the bacteria clear. Positive stain, the dye has a positive charge, it sticks to the negative cell wall of the bacteria, and the bacteria themselves have a color. Now that's great if we want to see the shape and how they're arranged. If we want to see more, if we want to know things about the cell wall of the bacteria, or if we're looking to see if the bacteria has a special structure that will help us identify it, we need to use different kinds of staining procedures. So here are the simple stains again. One dye is either a positive stain or a negative stain. Positive, the bacteria have a color. Negative, the background has a color. This middle panel here, we call these differential stains. Notice we're going to use there's two contrasting colors. It's easy to see in this this first panel with purple and pink, or purple and red, we say. In this one, it's not as easy to see, as well as in this guy, it's not as easy to see, but this one should be in sort of a greenish blue and red that you can see. So differential stain, we have two dyes, hopefully with contrasting colors, and they're going to tell us differences, that's why it's called differential, differences in the outer layer of the cell wall of the bacteria. And these are some examples: the Gram stain, acid-fast stain. Actually, in this acid-fast panel, you guys can probably blow it up at home. The little tiny red dots, I believe, are the acid-fast bacteria, and the blue cells, these bluish purple cells, are the non-acid-fast cells. Gram stain, #1 stain that is done every time in a clinical setting before that goes under the microscope. Every single specimen will be Gram stained. The acid-fast stain we only use for bacteria that we suspect have a big, thick wax in their outer covering. It's called mycolic acid. And this endospore stain, I'm going to leave that for a minute because it's actually an example of both a differential stain and a special stain. Special stains are stains used to look for special structures or unusual structures. Oftentimes the protocol is longer and or more difficult. Even if we were live and in person, you would not do any special stains. We would just look at some examples of special stains under the microscope that someone had already made for us, prepared slides. Here's an example of looking for capsules. You can see the bacteria in there are the purplish structures, the clear white around them, that is the capsule. So they have an extra coating outside of their cell wall. This is a bacteria that has flagella, all of these little wispy things that look like cracks in the slide. Those are all flagella for the bacteria to move around. And this particular stain is really difficult to do. So even the slides that we purchase, sometimes they don't look this good. And as I mentioned, the endospore stain is an example of both differential stain and special. Special because these tiny red cells here, those are the spores, and then the bluish purple cells, these larger ones, are cells that are still living and actively doing metabolism. So we can tell the difference between two types of cells, and it's also a special stain because not all bacteria can form spores. That's why it's kind of, I would have put that one here between the two, but not my book. All right, that ends, hit stop.